Thank you all for joining us. I am going to be moderating this panel, and the title is The Role of the Microbiome in Type 1 Diabetes, a walk through the evidence. And here with us today, I'm going to introduce you sort of one by one, is Dr. Emrah Altindis from Boston College, Dr. Mark Atkinson from University of Florida, Dr. Eric Triplett from University of Florida, and Dr. Johnny Ludvigsson from the Department of Biomedical and Clinical Sciences in Linköping University, Sweden. Colleen Cutcliffe is scheduled to join us and hope she will. She's a co-founder of Pendulum Therapeutics. So just a little bit about the panelists. The lab of Emrah Altindis, PhD at Boston College, conducts an interdisciplinary approach to address two complex and vital questions. One, characterizing the effects of viral insulin IGF-1, like peptides, VILPs, on insulin in the IGF system and host pathogen interactions. And two, determining the role of the gut microbiome and microbiome-derived metabolites in T1D and celiac disease. A recent collaborative paper with Dr. Ludvigsson continues to illustrate this realm, and we will drop that right now in the chat. So you have it there for your reference. Dr. Mark Atkinson doesn't really need an introduction. He's currently the ADA Eminent Scholar for Diabetes Research and the Jeffrey Keene Family Professor at the University of Florida. He's also the director of the Diabetes Institute at US, author of nearly 600 publications. He's beginning his 37th year of investigation into the field of type 1 diabetes. He's been the recipient of multiple scientific and humanitarian-based awards for these efforts. Dr. Eric Triplett is professor and chair of the Microbiology and Cell Science Department within the University of Florida's Institute of Food and Agricultural Sciences. His research interests include a particular emphasis on the gut and oral microbiome. His lab has identified bacterial associations with type 1 diabetes, autoimmunity, and premature birth. And Dr. Johnny Ludvigsson, MD, PhD, is a professor emeritus at the Department of Biomedical and Clinical Sciences. As I stated, Linköping University in Sweden. He specializes in clinical immunology, diabetology, and epidemiology. And current projects are immune intervention in T1D, epidemiology, and diabetes complication. He's been involved in a multitude of T1D focused clinical studies, including ABIS, which is all babies in Southeast Sweden. I just would like to take a moment to, thank you again, and then ask you, the format is that we're going to introduce, sort of the history of discovery of the microbiome's impact on T1D. And then, with Drs. Triplett and Altindis to lead that, and then, with others weighing in, and then talk about translational opportunities to the clinic, with Dr. Atkinson and Cutcliffe, and perhaps Ludvigsson joining in. Everyone is welcome to join in, and everyone is welcome in the audience to drop comments into the chat and questions into the chat. We'll field them as we go, and then at the end, we'll also have time for questions. So I will leave it now to the experts to take it away. Would you like to start talking about sort of the history of discovery?
I guess I can start with my biases. So I started this by introduction to Dr. Joe New in the College of Medicine. And he was interested in bacteria associated with diabetes and the diabetes-prone rat. And that became our first paper where we saw lactobacillus was associated with healthy rats. And then through that collaboration, Joe introduced me to Mark Atkinson and Des Schatz, who I've worked with, and who they introduced me to people from other cohorts, such as the DIP study in Finland, Baby Diet in Germany, and Yanni and Abbas in Sweden, as well as Teddy. And I'm now co-chair of the Infectious Agents Committee in Teddy. So if it weren't for Mark and Des, I wouldn't have any association with human cohorts. They got it all started for me. So that was great. But anyway, the long and the short of it is, we started by finding associations in the rat model, but then we moved quickly into humans after that. And through Ali Samel at Turku University and his colleagues there, I got associated with the DIP study. We found associations there and published them, particularly a strong association between Bacteroides dorii that preceded a seroconversion in the DIP study by several months in Turku, Finland. Very strong association. Then we connected with Annette Ziegler and her colleagues in Munich on the baby diet study, and we found bacterial network associations with controls that were absent in cases, so that the communities were quite disjointed in cases compared to controls prior to seroconversion. Then I got involved in TEDdy a bit. TEDdy, I was not a major player in their two major Nature papers, but they looked at the microbiome and children up to 4 years of age, all at genetic risk for type 1 diabetes, and found essentially no association with seroconversion in their two papers. They're quite disappointing. But then fortunately, Mark and Des introduced me to Yanni, and Yanni's cohort was special, the ABBAS cohort, all babies in Southeast Sweden cohort, because it's a general population cohort. It had subjects in it who were not at genetic risk, as well as subjects who were. And that allows you to study the host effect, the host genetic effect on the microbiome. And we've learned recent years that that's quite profound, both taxonomically and functionally. So that if you control for high-risk genetics in a cohort, you're missing out on a lot of the bacterial roles that could be involved in type 1 diabetes. So that's my preliminaries, and maybe go to Emrah from here. Emrah?
May I just add one comment? May I add a comment to what Triplett said? And that is, you know, when we started All Babies in Southeast Sweden, my idea and opinion was that if you have a strong genetic background, you don't need so much environmental factors or lifestyle factors. So if we really want to study how environment or lifestyle influences development of type 1 diabetes, then I would like to study those who do not have genetic risk. Or at least not a very strong risk. So that is the main reason why we wanted to have a general population. The most interesting in patients who do get type 1 diabetes are the ones, in my mind, who get it although they do not have a strong genetic background, because then it has to be very remarkable environmental factors. That was an idea.
Yeah.
Okay.
I think Yanni was quite right about that, because I think the reason TEDdy didn't find any bacterial connections with type 1 diabetes is because all the kids were at high genetic risk. So you're masking this tremendous effect just because of the genetics. I'll tell you one little anecdote that we're working on now. I've become interested with Yanni on ABBAS subjects who acquired type 1 diabetes without the high genetic risk alleles from HLA. And give you one example, we found an 11-year-old in that study who got type 1 diabetes at that age. And then at 1 year of age, that child had the most unhealthy microbiome I've ever seen. It was 78% Escherichia coli or Shigella or Bacteroides or Villanella, all these things we see in an unhealthy microbiome. And they had virtually zero of those microbiomes, of those bacteria we'd expect to see in a healthy gut. In addition, the mother smoked at home. She had serious life events, and he had infection after infection, all sorts of problems. So I think the inflammation required to get type 1 diabetes when you're not genetic risk is much higher than if you already are at risk. And so we think those are the cases where we can really learn a lot, where it's hard if you have control for HLA. Sorry, Mark, did you want to say anything before we turn to Emrah?
Yeah, before Emrah puts up his slides, and again, if anybody on the call has questions, please, I've got my chat open for any of us. Please put it in there. Again, I think just taking one step back, why do we think the environment is involved in type 1 diabetes? Is that if you look over the decades, the incidence of the disease, depending on the country, it's gone high, it's gone flat, or even in some recent studies, it's even declined a little bit. And this is before COVID. And if you look at the COVID literature, and this gets beyond microbiome, what the literature just says everything, that COVID either increased, decreased, or they had no effect on type 1 incidence. But again, going to Eric's point, so we have to come up with some reason why this incidence of diabetes rates fluctuates, and the best candidate we have for that is something in the environment. And we've done a really awful job over the last 3 decades of identifying any single factor in the environment, that allows for this. But then the only other point before Emrah takes off is again, what we do know 2 things about genetics. Number one is, over time, it appears that what in the 1940s and 1950s was a genetic risk for type 1 diabetes that was really high, to have the genetic risk seems to be declining over time, meaning there are more and more people in the general population that don't have this real high genetic risk, but do develop type 1 diabetes. So whatever that environmental thing is, it seems to be outpacing genetics. And then the second thing is, somebody has to figure out why. It's also increasing, that it used to be that 70 to 80% of people who develop type 1 diabetes had no family history of the disease. Well, now that seems to be eclipsing above 90%. So in a war between genetics and environment seems to be winning. And we need to figure out why. And the microbiome studies like those done by the individuals on this call are very important. Emrah.
Yeah. So first of all, it's a great pleasure to share the stage with you. So I first met with Mark, and he was very supportive. Mark introduced me to Yanni as a new investigator. Yanni has been also very supportive with the samples. This is also how we met with Eric. So I prepared a few slides for you. These are the points that Mark just also, I think, made. There's this immunological trigger, and we have no idea about what it is in type 1 diabetes onset. And because we don't know the trigger, we have no tools to prevent the disease. So currently there are 1.56 million Americans living with this disease, and the JDRF predicts that there will be 5 million people by 2050 in the US alone. And because we don't know this trigger, we cannot prevent it. So microbiome is one of the environmental factors that might be related to this trigger. And in addition, by using some microbiome-related tools, we can also try to stimulate tolerance after this immunological abnormalities happened to cure or manage the disease. There are several GWAS studies identified several factors that are related to type 1 diabetes. HLA is the first one, and Yanni Ludvigsson and Eric Triplett published a paper about how different HLA types affects the gut microbiome composition. However, these genetic factors are not sufficient to explain this increase in type 1 diabetes rates. This is coming from one of Mark's papers in Lancet. It is an old paper, but it shows this increased new cases per 100,000 people per year, Finland, Sweden, Colorado, and Germany. And also there are some geographical differences. As you know, Finland and Sweden are the top countries in type 1 diabetes rates, followed by Sardinia Island here in Italy. And these rates are increasing in most of the global north, not in the global south. So in the last 20 years, thanks to new genomics technologies and bioinformatic tools, our understanding of ourselves changed. Like when I look at these screens, I see one Homo sapiens organism in each box. However, we are much more complex than that. We are surrounded by microbes on all our skin, but also in our cavities, oral, nasal, vaginal, gut microbiomes are studied in the last 20 years. And now we understand that they are associated with different diseases, from cognitive diseases to cancer, infections to metabolic syndrome, type 2 diabetes, and obesity. And we also have been, working also in autoimmune diseases, specifically type 1 diabetes. A few years ago, we published this review to understand what we learned from ongoing gut microbiome studies and made this figure. Of course, it is very simple, but in healthy individuals, there is a thick mucosal layer to protect epithelial barrier from the gut microbes. in type 1 diabetes patients, this mucosal layer is decreased. One of the problems is that gut permeability is increased, so there is more inflammation in the gut. The diversity of the gut microbiome is decreased, and short-chain fatty acids are also decreased. The gut microbiome compositions are different, as Eric was one of the pioneers identifying Bacteroides. And then there are more inflammatory bacteria increased in the type 1 diabetes patients' gut microbiome. But the question is here, is it chicken or egg? So most of these studies are descriptive. So we don't know if they have different gut microbiome compositions because of the disease, because there are different stages of the disease, or there is a causal link between these gut microbes and the type 1 diabetes. So there is a real need to develop some mechanism-based, hypothesis-driven studies to understand a causal link, like the Koch's postulates, like microbe to disease or metabolite to disease as well. And we have been working on insulin-like molecules, but I will not talk about our study in these slides. The good news is that there are several studies, longitudinal studies going on in the world to address this question. So they have been collecting samples from several kids, like in the ABIS studies. Ludvigsson started this study, and they collected sample over 20,000 children without genetic risk, and over 100 of them developed type 1 diabetes. So this is a great, we have the biomarkers that we can analyze there. Diabetes study collected samples from Estonia, Finland, and Russia, and they collected these samples from 0 to 3, and they found some LPS-related changes in the gut microbiome that might be related with type 1 diabetes onset. I was very excited to learn more about the NDA study in Australia. TrialNet is going in the US, and TEDdy has been going on in the US and also with the European collaborators. And I went to the last IDS. This is where I was able to chat with Mark a bit. So I learned about this new study in Germany, GPPAD. I think the first study was baby diet. Now they continue to collect samples, but also organize some clinical studies to test some intervention studies on young patients. And INODIA is also another European study to test these samples. So lastly, in terms of how to use gut microbiome to treat the disease, I think these are two exciting new studies using prebiotics here to increase the short chain fatty acids, or genetically changing a Lactococcus lactis to stimulate tolerance in type 1 diabetes. But I will not talk about the details of these studies because of the time limitation. So I have one more slide about current limitations, but I want to cut here and we can discuss more if there are any questions.
I think one important point we need to mention is that viruses appear to be associated with autoimmune disease more and more over time. And that's certainly the case with type 1 diabetes. Teddy's big discovery in the microbiome was the association of enteroviruses with type 1 diabetes, although that was not necessarily new. Others had already shown that. But viruses do play a role, are certainly associated, but we don't know what the role is yet. But with that, I think that's our introductory remarks on what Colleen would like to add from her perspective, the corporate world.
Thanks for joining, Colleen. I'm sorry you had to struggle to join us, but I also just want to give a quick intro on Colleen Cutcliffe, PhD. She's the CEO and co-founder of Pendulum Therapeutics, and they have brought novel clinically tested microbiome products to the market. She has over 25 years of experience managing and leading teams in biotech, pharma, and academia. So thanks for joining us, and great to have an industry perspective as well.
Yeah, I'm really sorry I missed all of the introductory talks here. I was struggling with being let in as a panelist, but Monica figured out how to unlock me. I think I'm sort of coming from a pretty different perspective. So first of all, I am not a type 1 diabetes expert at all. And I would love to hear actually what everyone else said about it so I can learn more. Our company has really been focused on type 2 diabetes. And so I know there's a fair amount of overlap there, but there's also, some things that are different about that. And I think, in particular, some of the pathways that we've been working on where there's overlap are microbiome strains which produce short chain fatty acids, and then some of the microbial strains which are implicated in immune response and modifying the way the host immune response reacts to the microbiome changes. So, and then for us, kind of taking all the amazing science that people have done and being able to translate that into manufacturing strains at scale, as well as figuring out how to deliver them into humans, has been really the name of the game for us. And so we have clinical trial data in people with type 2 diabetes demonstrating that we can deliver our strains, as well as in a placebo-controlled double-blinded randomized trial, we showed that we were able to lower A1C compared to placebo. People who are on an intervention were able to lower A1C by 0.6 and lower postprandial glucose spikes by 33%. We have strains that were actually in the slide deck that was just shown that are in that formulation, including Akkermansia muciniphila, as well as 2 Clostridial strains and a Bifidobacterium strain and Anaerobutyric hilli. And so we actually have customers with type 1 diabetes who've been using our product and sharing back CGM, a continuous glucose monitor data with us. And so I think there's potentially some interesting stuff that we've done that might be tools that could be used by others. So I think that's really the kind of information that I can bring to the conversation.
It sounds like there's some room for collaboration here.
Could be. So where do we take the conversation from here, Monica? Where would you like us to go?
Well,
Yeah,
I mean,
Yanni has his hand up.
Yeah, he has his hand up.
Yanni.
So go ahead and ask something.
Panelists. I have my hand up because I had some comments or questions. It's interesting, of course, to start with regular trials and so on, but I have some naive questions, and that is like we are studying stool samples usually, which I suppose is the end of the gastrointestinal tract. It may show something from other parts, but I'm not the person knowing this really, because I wonder what role does it play to have a certain microbiome in duodenum or in ileum or close to pancreas, for instance. If it were so that certain bacterial strains were dangerous, went up in the choledochus and damaged the pancreatic tissue, it could play a completely different way, different role than just influencing the immune system. And I think also about viruses in stool, where in the upper gastrointestinal tract, or in the middle of it, or in the end of it, so to say. I don't know how much this has been studied really, and how much people know about that. I'm not an expert in the field, so I don't know, but these are the naive questions I think of. And then a combination of bacteria and viruses. How do they interact? Do they interact, or do they just live side by side?
I mean, those are all, I think, fair questions. And I will just start by saying we are very early days as a field in just understanding the microbiome and microbiome science for all the reasons that you just pointed out. So number one, we're typically measuring the stool. Sorry, my new Zoom gives me all kinds of animation. Number one, we are measuring the stool, which is a proxy for what's happening inside, but to the degree to which that's a good proxy is unclear. There are some technologies that have been being developed for a while to try to capture samples at the site and then deliver various parts of the GI tract data. I think those technologies or those pills are still in development, so it's sort of hard to tell. And then of course there's biopsies, which sort of have the advantage that you're getting the direct sample, but the disadvantage that if not handled in specific ways, you might lose some of these strains, especially because many of the strains that we're very interested in are strict anaerobes. Once they're exposed to oxygen, which is of course all around us, those strains die, and then you might not get to see them once you get to sequencing and certainly biochemical assays. So there's the sampling problem, and where we're looking, does that really represent what's happening? And that's very much linked to the delivery question. Where are you actually delivering these microbes to, and how can you know where you're delivering them? And I would say, there are enteric coated capsules, which help ensure that you're getting through the stomach acid. There are time release capsules, but they're kind of a one size fits all, and everybody's GI mobility is different. And so I agree with you. These are all kind of maybe make it miraculous that you could see any efficacy in humans at this point in our state of knowledge. And then lastly, not only, where are we sampling, where are we delivering, but then also how do these strains interact with each other, creating these metabolic maps where you're approaching this like a systems biology problem and basically saying, how do these microbes interact with each other? And then how do they interact with the host is still, these are maps with lots of big holes in them. But I think we do know some things that we can sort of see these strains which appear to kind of go up together and go down together. And then you can study them in vitro to kind of look at how they might be interacting with each other. So for example, we know Akkermansia is able to generate propionate, but it's not able to generate butyrate. But if you mix Akkermansia with various Clostridial strains, you can now get that secondary fermentation of the propionate into butyrate. And so some of our hypotheses are really derived from the fact that Akkermansia, needs Clostridial strain to actually complete the production of butyrate. And that, of course, butyrate is a super important small molecule, not only for colon health, but also for binding to G protein coupled receptors, and stimulating L cells to release GLP-1. So I think, it's still very early, and everything we know now will become things that will become very rudimentary or maybe totally wrong later. But, this is what we have to go off of. And for me, the most important thing is that we're, being safe about the way we do studies, but that we try to go into human as quickly as possible, because in vitro systems and animal systems are never great in general, but they're even worse for the microbiome because they don't really eat the same things that we do. And we know nutrition is such an important part of dictating, the microbiome. So I think it's really hard to make headway here without getting some human studies in. And for type 1, that's a huge challenge because these are kids and infants. And so there's, the safety part, the number one safety part maybe becomes harder to think about than when you talk about type 2, which is kind of in a, more established microbial setting.
We have a question in the question and answer box. Thank you for organizing this. Would panelists like to comment about other microbes in the gut other than bacteria such as fungi, phages, and archaea? And Teddy did publish on fungi and found nothing between case and controls, but again, it's a genetically restricted cohort. Phage, Teddy is also about to submit a paper to a prominent journal on bacteriophage they found in the gut. So I can't say much about that yet till it comes out. And I'm not aware of archaea in the gut. I think there's been much discovery there at all. I don't think they're common in the human gut. But anyone else who wants to comment on these things, have at it.
So I'll only say is, and again, I appreciate what everybody's saying here. I think when the microbiome studies first came out in type 1 diabetes in the mid 2000s, 2007, again, there was great optimism that this was, the new frontier and answers were going to come fast. And I think if we look back, we as a community didn't do a few things very well. Number one is there's the microbiome. What are we talking about? Are we talking about pathogenic mechanisms? Are we looking at the microbiome being involved in pathogenesis? Are we talking about biomarkers, disease biomarkers? Are we going, is that our goal here? Or as Monica said in her intro, therapeutics. And I think these things all kind of got intertwined, and we haven't had the best of pathways in terms of focused questions. The second thing, I think we were naive a little bit, and Eric published a paper, and I know Yanni and others, we didn't, Colleen, you mentioned childhood and infancy. I think we did not take into account early life dietary influences, antibiotic usage, et cetera, in terms of populations. And like I said, Eric's published on this, and Teddy, that which is a confounding factor. And then finally, and then maybe Emrah wants to comment on this, I think ultimately animal models are going to prove valuable in terms of mechanistic findings. But I agree with you on a direct day-in-day surrogate with genetics and then a fixed diet versus environment. There were some challenges there. Emrah, what would you respond to that?
Yeah, so I totally agree. The one point I prepared one slide about the limitations. Like I think it is related to this discussion here. So let me. So I think that the current limitations in the field is, yes, everything is gut microbiome centric in the type 1 diabetes research. There, I haven't seen any oral microbiome. Like I think that the NDS study is now working on the mother's vaginal microbiome, but not from the patient's nasal or skin microbiome. Everything is very gut microbiome centric. And all gut microbiome studies, or most of them, are bacteriome centric. We have very few virome studies. One was published in the PNAS. Teddy recently published one paper, but still there are very few papers there. Yeah, phages, you just mentioned, Eric, that this paper will be published, so I'm very curious. Everything is DNA centric. DNA means that who is there, which microbes are there, but I haven't seen a proteome paper, except Emma Hamilton just published a paper last year on proteome and metabolomes. But we need more papers to see which proteins are there or which metabolites are circulating or in fecal samples. We have a diversity problem in the type 1 diabetes field because most of the studies are coming from either Europe or the US. There are very few studies coming from or samples coming from other parts of the world. And as we have been talking about, these studies are mostly descriptive and very few studies have mechanism. In our recent study that we collaborated also with Mark, we tried to focus on one molecular mimicry mechanism and identified one bacterium. But I think we need more mechanism-based studies. So these are the main limitations. And I have something related. I have been receiving emails from type 1 diabetes patients, relatives, like the parents, asking about probiotics. Should we use probiotics? So when you go to CVS, you will see all these different marketing, so many different kinds of probiotics, but there is no science, I think, behind them. I don't know if they are still alive or not, this bacteria. And just before this talk, I checked what is the market. Like in the US, it is $17 billion, all these probiotics. And I have no idea if they really do anything. I don't know if they even survive their stint on the shelves of the CVS.
I think Colleen's group was working hard to solve that problem because you're quite right. About a decade ago, I had an undergrad in the lab where we just bought a bunch of probiotics off the shelf and asked the question, what's really in them? And what was in them had nothing to do with what the label said. And I didn't publish it because I didn't want to get sued by one of these companies. I didn't want to have to worry about that. But I believe quality control is changing a lot with groups like Pendulum. Colleen?
Yeah, I think there's a lot to be desired on the regulatory front here. And I will give you an example. I mean, Akkermansia, we have like 22 patents issued around the use of this in metabolic syndrome and even around, manufacturing and things like that. And on Amazon, now that people are starting to know what Akkermansia is and they're looking for it and seeking it, there are Akkermansia products popping up, and we call them Fakermansia because we buy them, we send them to third-party testing. Obviously, we can sequence them ourselves as well. And there's just lactobacillus in the pill. And so the pub, and we just pulled 5 last week. So the public is really at risk of, deception. This has been, you're right, it's a $17 billion market, it's a $70 billion market, $70 billion market globally. And when you have those kinds of numbers where people believe their gut health is important and they're trying to figure out how to enhance strains that are going to be beneficial to their health, but there's no real measurable impact to that. So they don't know if the thing's working or not. When you live in a world like this, it is ripe for all kinds of charlatans and frankly, people selling snake oil to make their way in. And here in the US, anybody can sell anything, and the FDA and the FTC are really in a backlog kind of way. They cannot proactively go after these, and so it becomes
Is
really
this,
hard.
Colleen, is this due to the fact that these supplements are considered food? Is the regulatory relaxation around it, or?
Well, they're considered supplements, and so dietary supplements are not, unlike drugs, they don't go through the FDA for pre-market approval. Anybody can slap anything onto a website, and apparently onto Amazon. So, I think this is a problem. I think it would be. There have been a couple of academics who have published on kind of truth in the bottle, or what are you actually buying. I hear Eric's sort of trepidation around backlash, because these are companies that have money, but it's a real struggle. And it's going to continue to be, I think, especially here in the US.
Yanni, did you want to ask a question?
Yeah, I wanted to come in just to comments. One thing in relation to what Emrah said about limitations and different parts of the world, it may be so. I don't know. I just say it may be so that what is protective in a certain area of the world is not protective in another area, and what is dangerous in one area is not necessarily dangerous in the other. I don't think there is one single thing like a virus or certain bacteria or something which has the same effect everywhere and causes or protects against type 1 diabetes. So therefore, that's one thing. It may differ geographically what we see and what is the result. But and then another thing regarding what we are talking, which I think Mark was into, and that is, are we talking about preventing the process to start early in life, to stop the immune process? Are we talking about gut barrier function, antigen presentation, that sort of thing? Or are we talking about, which may be another thing, when the process has gone on for quite a few years and you are getting closer to precipitation of the clinical disease, what role does it play then, and so on. So it may be different things. It may be so that certain microbiome may be either protective or dangerous very early in life, but not necessarily the same 10 years later. I'm just provoking, giving some questions.
So building on this, thanks, Yanni. Building on this conversation, I think things are getting worse and not better. There was an article in Wall Street Journal within the last week talking about Wegovy and Ozempic, and sorry for mentioning trade names. But again, you can go online now, like Colleen said, and there's, I think, at least 60 companies that sell what they claim are biologic equivalents, for a cost here. And I think this is just the future. You go on Facebook, Instagram. I get AARP. I'm old. And, there'll be, ads in there for biologics in there too. I mean, it's something that we're going to have to deal with. I don't know exactly how. Yeah, and we don't seem to do a great job. One thing that we haven't talked about, it wasn't on your discussion list, that in terms of the evolution in the microbiome in the last 15 years in type 1 diabetes, and it related back to the initial studies in animal models on obesity, where people were talking about fecal microbiome transplants, meaning the original one, fecal microbiome from obese mice to lean mice. And there was a push for a while that this was going to be the future in terms of type 1 diabetes. And I think, based on other medical conditions that ranging from autism to type 2 diabetes, obesity, one of you want to talk about fecal microbiome transplant? We talk about nutritionals, but where do you think that stands now as a therapeutic?
My sense is that works extremely well for a serious gut problem like chronic diarrhea. It's called by Clostridium difficile, right? For that, it works really well. But I haven't seen much of it working for other ailments, but I haven't looked recently. Does anyone else have anything to add on that?
I think there are studies ongoing in Holland, in the
Yes.
Netherlands,
Yes,
if I remember correctly.
that's true.
And in cancer research, they also identified that the gut microbiome composition determines the response of the patient to some of the cancer drugs, like the checkpoint inhibitors. But back to Mark's point in type 1 diabetes, unfortunately, I haven't seen any FMT study that worked in NOD mice or in humans. So this is one of the limitations. But as you know, we were able to identify one bacterium that was able to increase the type 1 diabetes rates, and people identified some that can prevent. But you wrote a lot about this, like we were able to prevent type 1 diabetes more than 300 times. Mark, this was your sentence, in NOD mice, but we were not able to transfer these prevention studies to humans, except one that delays. Right?
Yep. Monica, can I ask the group a question?
Of course.
All right. So if you were going to, and again, maybe brief answers because we're running, I'm looking at the clock here. If you were to say, if you had $100 million to spend, would you put it on looking at the role of the microbiome in pathogenesis, therapeutics, or as a biomarker for identifying disease risk? Now, I mean, just going around, where do you think, where would you place your bet moving forward? Yanni?
Good question.
Do you ask me?
Yes. I'm asking everybody.
If
but
I
I'm
should
starting with you.
put my money, I would do it in the to find the start of the process early in life, to see whether you in some way, either whether it is on affecting the immune system and or gut barrier and antigen presentation. That first year or 2 years of life, I think when the process starts, that's where I think microbiome plays the most important role. And then it could very well be so that certain parts of microbiome could be protective and others could be causing, contributing to the disease. And here we have both viruses and bacteria, so we have to look upon both, I think.
So I take that as a vote for pathogenesis. Emrah, where would you put
I would
pathogenesis, biomarker, or therapeutic?
Yeah, I would definitely put half of the money to understand the trigger, because if we can understand the trigger, then we might think about some tools to prevent it, as I already said. But I'm also very excited about the genetically engineered bacteria to stimulate immunotolerance in the gut against some autoantigens. There is this new paper, IL-10, is by Lactococcus lactis, overproduction of IL-10. This is also studied in other also autoimmune diseases. So if we can also help type 1 diabetes patients by stimulating immunotolerance, it would be also great. So 50-50, Mark.
Eric?
I put in therapeutics with the eye of getting a collection of strains that can reduce inflammation in early life, but paying particular attention to each child's genetics, because that makes a huge difference on what can colonize and what can't.
And Colleen? And not that I'm in charge, but I
No,
would.
it's good. It's great.
I really like the opinions here. Colleen, how about you?
Well, we have raised over $100 million and we put all of it into therapeutics. And I think we have the luxury of leaning on all the great work that you guys and others do in order to understand more about pathogenesis and biomarkers. But the reason that we've gone all in on interventions is because I think in this space, getting longitudinal data on the microbiome and then perturbing the system with something that you kind of know exactly what it is, which is sort of the difficulty with fecal microbiome transplants. But if you have a defined, consortia, you perturb the system, and then you can understand what change did it make to the microbiome, and then you are measuring health outcomes. That for me has been kind of our path forward in the space. And we haven't done anything in type 1, so would love to talk to any of you guys who might be interested in that kind of a model.
Yeah, just to sort of build on the back of that question, and we talk about like how.
Wait, are we not going to get to hear what Mark?
Oh yeah, Mark. Okay, okay, Mark. Since you posed it, you can talk.
I'm looking at retiring at sometime soon, so I'm not.
No.
Although I aspire to be like Yanni in one way, again, just keep pushing forward. I actually like the two. I like Emrah's answer. I think which combines everything. I just don't know, given what's been done about the biomarker side, even though a lot's been done at that. The literature is so conflicting in based on populations. So I like that notion of Emrah of a 50-50 split between it, which is all your answers. Let's try and figure out how this can influence disease pathogenesis, but at the same time, take advantage of that smart and try and design therapeutics around it. But thanks, Colleen, for asking it. But I'm just repeating what you all said.
Well, okay, so I think those were great answers. I wondered, just to build on the back of this and kind of tap into what Emrah brought up, is how do you sample in young children? How do we get a understanding of the landscape, of the clinical landscape? Is there a way to build this kind of microbiome sampling into well baby checks? As Yanni said, want to go for the sort of the youngest clinical set. Is it possible to even try to, as Emrah was talking about, maybe start sampling the oral microbiome or, other parts of the microbiome? What are your thoughts on that?
I'm keen on oral microbiome. We published a paper not long ago on how it can be associated with mental health. And it is the first entry of bacteria into the human body. It's relatively easy to sample. And we published a paper recently where we thought oral microbes are far more associated with pancreatic cancer than gut bacteria are.
Mm-hmm.
So I think that's an understudied place, even though it's so easy to sample. But I also think that we need more about host genetics and how that interacts here. And we do know what an unhealthy microbiome looks like. So in young children, we could identify that. And if we know their genetics, that helps even more. But in some cases, you're more likely to see a less healthy gut if they don't have at-risk genetics. So I think there's a place for that.
And what would be the steps to incorporate that into the clinical sampling? Or, you have to get the, pediatric ADA or something behind that, or what's the step?
It's not easy. There isn't even a consensus yet. Mark will know better than me. There isn't a consensus yet on even genetic screening of children at birth, right? So, and that's easier.
Yeah.
Emrah.
So one of the people in the audience reminded one FMT paper. So there was a study from Netherlands and Italy, a collaborative work that they did FMT transfer to 10 patients. So the sample size was low, but they showed that these are the early diagnosed new onset patients. They were diagnosed, the treatment starts within the 6 weeks after diagnosis. And they make this FMT transfer in the first 4 months. And they show that endogenous insulin production was decreased. So they were able to preserve the beta cells much better than the control group. So there was a 2021 gut paper. I apologize that I forgot this. There are also some questions in the Q&A.
Yes, I'm looking at this now. Are there any trials collecting samples, milk, feces, vaginal from mothers of type 1 diabetic patients? I'm not familiar with that. And Dia is doing something close, but
yeah,
I think Dia
in Australia, right?
Yeah. I think Dia is as close as we get. You're right.
Right. And then let's see, is the translation?
Sorry, I just wanted to ask a question that's maybe related to some of the Q&A, but also the topic here. How do you guys on the panel, how would you feel about these sampling from different sites, baby and mother, over time, if it was done not through a clinical trial, but as like a direct to consumer? We can think about, 23andMe, people are spitting into a tube. A lot of people have done that. And I think parents, speaking as a mother myself, are invested in the health of their children and might be more willing to do a lot of these things than maybe a physician who's unsure about what they're going to do with the data. I'm curious what you guys think about that kind of a concept.
I think there's great potential for that. In fact,
Yeah.
one of my postdocs and I are talking about a service realm we could put up for that very purpose. I think you're quite right.
But to make I think sample collection also needs to have some glycerols, because in these tests they are just interested in the DNA, but you might be also interested in the bacteria as well coming from the oral sample. So it should have also some glycerol. Also, when collecting fecal samples, it is important to add some glycerol as well.
In the Q&A, is the translation difficult because of the genetics makeup of the individual habits or the place they are living? What are the most significant challenges? I presume they mean translation from animal models. I don't know. What are the challenges there? I just think it's a different critter. Mark, sorry.
Maybe clinical translation.
Right.
Yeah. I have an answer to the Colleen's original one, but I want to hear from Yanni first. Yanni, did you want to answer Colleen's question, or you want me to go?
No, do that, please, because I'm not quite sure I got it.
Okay, so I think, again, when I say that this has been a disappointing field, unfortunately, every dietary study seeking type 1 diabetes prevention, that where the mechanism has been presumed to either act on a microbiome or an immune modulating area or gut leakiness, has failed, be it breastfeeding versus bottle feeding versus casein versus hydrolyzed casein, cow milk avoidance. I mean, vitamin D, all as a singular agent. These studies have not met their endpoints. So clinically, it's been hard to push. But I love Emrah's slide in that I think these things are just taking time. We now have general population screenings, is becoming more and more predominant, as Emrah's slide showed. And I think now with at least one FDA-approved agent out there with teplizumab, now at least we have something that we can offer. And I think my hope, and I know Yanni's leading a number of these trials, is that over the next few years, we see more and more of these drugs approved, either by Emrah or FDA. And when that happens, I think people will participate more and more in these trials, and then allow for more studies in the microbiome. Maybe that's a wish list.
Yeah.
But
and in Jane Daska's recent paper came out last week, she showed that bacterial associations are related to this drug that delays type 1 diabetes, and particularly works well in DR4, not DR3, pointing again to the role of host genetics. So I think Mark's right, Emrah's right, everybody's right here. We're in for exciting times going forward.
Let's see here. We're talking about the therapeutic use of gut microbiome, but recent studies show that SCFA, the fatty acids, short chain fatty acids produced have adverse effects. How can we really overcome this issue?
I don't know which study they are talking about, but Emma Hamilton in Australia, they have been working on some different fiber diets that increases the short chain fatty acids, and they show that it didn't have any safety issues. It has some GI related adverse effects, some small ones, but nothing like related to unsafe conditions.
Yeah, we've had her showcasing her work, and I hadn't heard about adverse effects. Okay, Daria Ignason, I'm using Anna Cassu's link. Can Dr. Triplett please describe what characterizes an unhealthy gut microbiome in children? Point us to any relevant publications. Can the same be said about the adult gut microbiome?
I don't know enough about the adult, but generally speaking, and this has been known for a long time, that where we see a higher ratio of firmicutes to bacteroides, it's generally healthier. So in the case study I mentioned earlier, we see a lot of Escherichia, Shigella, Villanella, and what was the other one? Bacteroides in the gut, and very few Bifidobacterium, Lactobacillus, Collinsella, Coprococcus, etc. So we more or less know the good guys, the bad guys now. And I think we can certainly pinpoint a very unhealthy gut. But we got one more paper come out soon to show that. One other frustration we have is that Although sometimes we see a lot of butyrate producers in the gut, that does not translate to more butyrate as a metabolite in the gut. So that could be just a higher turnover there, what's going on. But these things are difficult to parse out.
Butyrate's essential for differentiation of naive T cells into Tregs, right? So, so is that not happening?
I don't know.
Can't really see.
Just,
Well, I think
my observation doesn't necessarily mean butyrate isn't working, it's doing its job. It's just hard to see that necessarily show up in a higher concentration. That's all it is.
I think this gets back to one of the earlier comments about sampling. So we've also found sometimes it's hard to see higher butyrate levels in the stool, maybe because butyrate is such a coveted small molecule that as soon as your microbiome is making more of it, it's immediately consumed by all the cells, and so you don't see it come out in the stool. So what we found is actually more sensitivity in plasma measurements of butyrate, and so that might. We've seen with our products it go up in both stool and plasma, but it's a larger dynamic range and signal to noise ratio in plasma samples.
Which is a whole other entity that has had little discussion on this. Otherwise, extremely great call, and Monica, thanks for organizing it. Let me just say that. But how is the microbiome acting here? Is it immunological? Is it working on gut leakiness? Is, what are the influences here in terms of how it's acting? And, I think that'll be the subject for future studies.
Or all the above, right?
All of the above.
Yeah, it is interesting. I will just comment that we had a talk a week ago with Ernesto Nakayama at PNNL. And he was looking at, they were able to look at 50 microliters of plasma and using site exclusion chromatography, run plasma EV enrichment for biomarker studies. And he indicated that, what they were seeing, really sort of elegant studies that, basically fatty acid processing was messed up from the beginning in some of the type 1 patients they examined through Teddy. So that was kind of, right. So that could be sort of the SCFA, component, who knows? I mean, it's really interesting work to be done.
What Yanni and I found in Abbas is that the set is staged for inflammation very early in age, at a year old, and or even in cord blood. And Yanni and his colleagues have published that. So, and then it's how that's modulated by HLA. So I think we can catch it early. I think that's the way to go if we can.
Well, my bet's on Colleen here, leading the charge into these waters, because she's done a great job with Pendulum and bringing things to market there. And I'm hoping that, these conversations will continue, and perhaps she could, afford an avenue into that realm with the help of all of you experts. So in the interest of time, I know Mark had to hop. Thank you all very much for joining. This was fascinating, and especially so late at night in Sweden. Thank you, Yanni, for joining. And
Thank you very much.
And we record this, and hopefully we'll spread the word, and people can reach out directly. Thanks again. Have a great rest of your day.
Bye, Monica. Thank you.
Bye-bye.